Year 11s. This is for the highest students only. We're going to go through the highest of B2. I just want to show you the weather from my front window. Can you see? This is always an opportunity for science. Now, the weather is... I quite like the rain, so I'm not going to say it's miserable. It's amazing. However, there's no sunlight. We've got lots of water. The amount of carbon dioxide is the same. What is the limiting factor here, people, and why? Think about that. And we'll go through the answer in a bit, so think, what's the limiting factor for photosynthesis? That is correct. The limiting factor here is the amount of sunlight and the amount of heat. Because the sunlight is used by the chloroplast to take part in photosynthesis, and the good amount of temperature is needed. Why is it needed? Because the enzymes will work well. Now, what we're looking at today is the higher things from biology only. Okay, so we're going to start off with the Human Genome Project. Now, with the Human Genome Project, what they've done is they've managed to map out every single gene within the human DNA, which is amazing. Right, so, and what they found is that every single one of you, if you look around, if you look at home, if you look wherever you want at human beings, we all share 99.9% of our DNA, which makes sense, but it's still amazing nonetheless. Now the DNA, what you got to think about the Human Genome Project is, well, so what if they've sequenced the DNA of humans, so what? And you got to think about it from the point of view of DNA and what it can tell you. Now, number one, there's an amazing video, I think it's called the Gene Project on YouTube, and basically what it shows is that there's no such thing as a pure race, okay? And that's kind of linked to the first thing, which by looking at and knowing your genome sequence, you can trace back your ancestry and you can trace back the evolution of human beings. So that's point number one. So point number one is you can look back to the evolution of human beings. Now, number two, you can study specific genes and look at how they can cause diseases, because that's quite important, isn't it? Number three is you're looking at gene therapy. And this is linked with number four. So you're looking for personalized medicine. And gene therapy is basically, you're looking at faulty genes, and you are looking at those faulty genes and you're kind of saying, well, what's wrong with them? Can we fix them? And so you're targeting specific areas to cure people. Now, this is the future of medicine. Okay, this is really, really exciting times, and you're going to be studying the genes. Right, you're going to be studying things that cause Alzheimer's, that increases your risk of diabetes, that increases your chances of developing heart disease. Then you're going to be targeting those genes specifically for specific people. And you're going to be switching them off, modifying them, doing all these kind of things to increase the lifestyle of people, right? And this is quite a recent medicine. Some have been quite successful and they've dealt with people quite well and cured diseases. However, some people have died, unfortunately, in the process of gene therapy. But of course, that is what medicine is. It's trial and error until you get something right. So that's the Human Genome Project done. We're now going to be looking at clones, making clones. Okay, so pause this. If you got any questions, you can ask or make some notes or rewind it, whatever you want to do. Okay. So here, this is about cloning, guys and girls. Now, if you look at this, this cat looks wild chuffed because he's getting a clone done of himself. He's like, yeah, I'm the cat, I'm the cat. So what happens with cloning is you take a cell from this cat that you want to clone, right? So that's the DNA, that's the nucleus, and I'm just marking it with a different color. And you're going to take the nucleus out of the cell. That's the first thing. Now, the second thing is you're going to take an egg cell, right? And I've left this one blank so that we can know the difference between the nucleus of the cat that we're cloning and the nucleus of the egg cell from the surrogate mother, right? Now, what you're going to do is you're going to take out the nucleus from the egg cell. This is called enucleating the egg cell. Okay, so we're going to enucleate the egg cell. We're going to take the nucleus out. Then what we've got, so we've got the nucleus from the cat, which contains the DNA. It's a diploid. You got to remember it's a diploid, so it's the full number of chromosomes from the cat. So we've got the nucleus from the cat, and we've got an enucleated egg cell. So we're going to add the nucleus into the enucleated egg cell. And happy days, you are on your way to making a clone. Look how happy he looks. Well chuffed. So once you've placed the nucleus into the enucleated egg, the next thing you're going to do is you're going to stimulate it with an electrical impulse. Okay, so this is my electricity going into it. Once you give it an electrical stimulus, it's going to divide by mitosis. Okay, so this egg cell with the new nucleus from the cat, diploid, remember, is going to undergo mitotic division. And you're going to place it into the surrogate mother's uterus. Now, this surrogate mother doesn't look too happy, doesn't know what's going to happen, a bit weird. And then, finally, after the duration of the pregnancy, and once the full term is carried out, the clone will be produced. Brilliant. Okay, so this part is about transcription and translation. Now, transcription means to write across. What are we writing across in particular? The DNA. So here I've got an enlarged picture of the nucleus. So we're forgetting everything else, we're just looking at the nucleus. And we have a double helix DNA. And what's going to happen is an enzyme is going to unzip the DNA by breaking the hydrogen bonds within the DNA. So what you'll have is your DNA unzipping to expose the DNA nucleotides. Now for DNA, the nucleotides are adenine, thymine, guanine and cytosine, or ATGC. A and T are complementary and G and C are complementary. Now just worth noting, as we just said, these are usually held together in place by weak hydrogen bonds. A and T have 2 hydrogen bonds. G and C have 3 hydrogen bonds. The way I remember that is AT has 2 hydrogen bonds. Listen carefully. AT has 2 hydrogen bonds. AT2, AT2, AT has 2 hydrogen bonds, and GC therefore has 3. Now, once it unzips, the DNA unzips, mRNA comes and lines up next to its complementary DNA. mRNA stands for messenger RNA. So the question is, why do you need this mRNA? Why don't you just get another set of DNA? Because that'll make life easier. Because mRNA has complementary bases of adenine, uracil, guanine, and cytosine, AUGC, where A and U are complementary and G and C are complementary. So why do you need mRNA? Why can't you just have another strand of DNA? It's because DNA is too big to leave the nucleus, whereas mRNA is small enough to leave the nucleus. So A will be complementary to U, T with A, G with C, C with G. So I've just done a short strand just to show you how it matches up. Now the mRNA, like I said, is small enough to leave the nucleus. And because we've just written across the DNA, we've just done transcription. That's transcription done. We've written across the DNA using mRNA. And the mRNA leaves the nucleus because it's small enough it can. And now we're doing translation. So we're going to take this mRNA, this messenger RNA, and translate it into a protein. We're going to read the message and make it into a protein. So a couple of keywords that you need to know. So I've just taken my chain and I've made it longer just to show you guys what's happening. Now these here are called tRNA, right? Each tRNA is called a codon. So this one here is a codon. Right, so what we have here is a tRNA. A tRNA is made up of 3 bases or nucleotides. Now each tRNA will have an amino acid. So this is one amino acid, right? The next 3 codon will have another amino acid. The next 3 will have another amino acid. the next three will have another amino acid, and these are carried by these tRNA molecules here. So tRNA stands for transfer RNAs. Right. So the transfer, now this process of translation, that is reading the mRNA to make protein, happens in the ribosome. So the mRNA goes into the ribosome, the complementary tRNA codons match up with the mRNA, and then what happens is you'll have a chain of individual amino acids. Now these individual amino acids will eventually fold up in a unique shape to make a protein, and that's translation done. So again, I just want to recap. Translation is when you've got an mRNA in a ribosome, right? Every 3 tRNA molecules is classed as a codon, right? When the codon lines up with their complementary mRNA, you end up having a long chain of amino acids, one after the other. Now, the long chain of amino acids end up forming bonds, as you can see here, right? And these bonds are peptide bonds, which are bonds that form between proteins. Now, once you've got the long sequence of amino acids, they will end up folding into a unique shape to form a protein. So we've gone from DNA to mRNA to tRNA to amino acids to proteins, and that is transcription and translation, and that proves and that shows how DNA codes for proteins. So if there is a mistake in the DNA sequence, there's going to be a mistake in the mRNA sequence, which means there's going to be a mistake in the translation process, which means there's going to be a mistake in the sequence of amino acids, which means there's going to be a mistake in the protein, which means the protein is going to be in the wrong shape, which means that the protein will not work properly. And that is what a mutation is. Okay, so this is from your textbook. Now, I've already covered every single part of the digestive system in one of my B2 videos. It's from November 2012. Now, on my YouTube channel, I've written a description, and in that description, it tells you where the digestive system one is. So I'm going to suggest that you look through that to get a rough idea of what everything does here. However, for the higher bit, what you need to be aware of is point number 9. So I'm just going to zoom into number 9. It's on the liver. That is called the gallbladder. Okay. Now, the job of the gallbladder is to make bile. So, well, store bile. The liver, number 8, actually makes the bile, and it's stored in the gallbladder, number 9. Now, an interesting fact about the bile is that it's what makes your poo brown, your feces brown. Isn't that amazing? Absolutely amazing. Now, the bile has 2 jobs. Number 1, it's alkaline. So, as the food comes out from your stomach, it's going to be quite acidic. It's going to go into your small intestine. Now, this is where the bile comes into play. The bile is alkaline, so it's going to neutralize that acidic food that's gone into your small intestine. So that's job number 1, neutralizing the acidic food that comes out into the small intestine. By this time, it's actually called chyme. It's obviously not food, because you wouldn't eat that again, I hope. And the second job is that what it does is the bile will emulsify fat. That means it's going to give it a large surface area. It's going to break it down into smaller pieces, which means that it's going to give it a large surface area, so the lipase can break down the fat into 3 fatty acids and glycerol at a higher rate. So those are the 2 main jobs. Number 1, neutralize the acidic chyme, and number 2, increase the surface area of the fat by breaking it down, and therefore the lipase can get to it, and it does this by emulsifying the fat. Okay, so that's that done. Now this again is from your book, and we're looking at the small intestine and what the job is. So this is again just for higher students. Now the villi is amazing, okay? It's found in the small intestine, and the adaptation that it has is absolutely amazing. Now, number one, if you think about it, it needs to be thin so that digested material can get through it. Because it's thin, it's going to allow only the small molecules to get through. The large molecules will not be able to get through, which is why there will be some enzymes within the small intestine so that it can break down food further. So this large molecule here cannot get through. It needs to be broken down by appropriate digestive enzymes so that it can get through into the blood. Now the second thing is that it has a large surface area, the villi within the small intestine. Why does it have a large surface area? So that it can absorb more nutrients into the blood. Next, it's got a very, very good blood supply. The reason why it's got a good blood supply is because it needs to carry away all the digested and broken down parts of the food. So it's got a good blood supply, large surface area, it has thin walls which are 1 cell thick. Okay, cells which are 1 cell thick, which allows for absorption of the broken down food. And obviously this occurs by diffusion, because if you imagine, right, if you imagine here, this bit here, I don't know why I got a potato peeler in my room, but I have. If you've got this in high concentration, the blood is going to take away whatever it's already absorbed, and so there's going to be a low concentration within the blood. So if you imagine this is the inside of the intestine, that's the villi and that's the blood. So the broken down food is going to go from a high concentration to a low concentration, and this is called diffusion. Right, so the nutrients that's been broken down, the food, will diffuse into the blood. Okay, now some people suffer from celiac disease. Okay, the spelling for it is there, spelled a bit weird. Okay, now if you look here, this person here has a lot of damage in the small intestine, right? So what you're going to have is you're going to have a lower surface area compared to this one. So if you've got a low surface area, you're not going to absorb as much. If you've got a low surface area, you're not going to have enough blood, so your diffusion isn't going to happen as well. And because the intestine has so much damage, the villi has so much damage, it's not going to absorb so many things, so you're going to lose more of the good stuff that you need. So you're going to lose your sugars, you're going to lose your proteins, your amino acids, you're going to lose your glycerol, you're going to lose your lipase through feces. And so what this means in turn is that you are going to lose a lot of weight, you're going to be feeling very tired, you're not going to have energy for respiration because you don't absorb the sugars, you're going to have some muscle wasting if you don't take enough protein because your amino acids aren't there. So you're going to have a lot of health problems, and this is evidence for the importance of the small intestine, aka the adaptations, the villi, etc, etc. I hope you got through this video. Come stai? Kif halak? Yakshimash? What else do I know? Chifach, and finally Kek Dela. Goodbye.